Thank you very much, Jette, and good afternoon, everyone. My name is Lykke Stylo, and I am Associate Professor at the University of Copenhagen, and it is my great pleasure to be the chair of today's double seminar, where we start with Steffen Raun, and then after that we have Dr. Henning Langer as well. So, but first, I will introduce Steffen. He's a postdoc at the University of Copenhagen, and he will talk about cancer metabolism and AMPK. So welcome, Steffen.
Thank you very much. And thank you so much for the DDA to allow me to present some of my work. Hopefully my sharing works as well. And I'll hide the panel. Everything is good?
Yes, everything is good, Steffen.
Great. Today, my name is Steffen, and I'm a postdoc at the group of molecular metabolism in cancer and aging here at the University of Copenhagen. And today I want to talk about how cancer affects glucose metabolism and the role of AMPK. Yes. And now everything is crashing. It doesn't change on your screen, no?
No, but maybe try to close it down and open it again. Sometimes that works.
Okay. Share.
Yeah, I think
we're good.
I've your second slide, Steffen. So
yeah, it's fine.
Try and
it
jump
doesn't.
Forward once.
Huh?
Maybe try and jump forward or backwards just once to check.
Very interesting.
Okay,
I could go forward. That's what I can do.
Okay, but that's good. There you go.
That's fair enough. We'll make it work. So today I'm gonna talk about, or I'm gonna give a brief introduction into cancer and how it affects metabolism. Then I'm gonna talk a little bit about what we've shown previously and what we've published before in our group. Then I'm gonna show some new data we have on muscle AMPK and metabolic dysfunction in cancer, and then some concluding remarks. And before actually jumping into my main topic of the talk today, I do want to state that there's also this other relationship that metabolic dysfunction and obesity there is an increased prevalence of cancer. So this is something you have to bear in mind, but it's not something we're going to talk about today. We're going to talk about how patients with cancer can develop metabolic dysfunction, and this leads to an increased cancer-associated mortality. So if you see over here on your right, this is non-small cell lung carcinoma, or patients with non-small cell lung carcinoma. I'm going to talk a little bit more about this cancer later. But if you look at the blue line and the green line, so this is the overall survival probability of the patients. And in the blue line, they have separated the groups into a fasting plasma glucose below 7 millimolar, or in the green line, if they have a fasting plasma glucose that are higher than 7 millimolar. And you do see this quite marked effect of if the patients have elevated fasting blood glucose on the overall survival probability. So there is definitely important to consider these metabolic dysfunctions in patients diagnosed with cancer. So, but what is cancer and insulin resistance really? So here, this is a older paper from '87 where they made a hyperinsulinemic euglycemic claim. to investigate the insulin sensitivity on a whole body level. And this is healthy controls, where you see as the x-axis, that's increased insulin concentrations. And on your y-axis, you have the glucose disposal on a whole body level. So how well the subjects are responding to insulin in relation to glucose disposal. And you see with increasing insulin concentration, then you also have an increased glucose disposal. Then down here, we have colorectal cancer patients, and you do see this downward shift. So when you look at the biological response to, if you look at the half maximal glucose disposal, you actually need quite a bit more insulin to get the same biological response if you're a patient with cancer. So there's a right shift and development of insulin resistance in these patients. And now I'm a molecular physiologist, and I'm interested in what's going on in the specific tissues. And this is a busy slide, but you don't have to read all of it, so to say. I just want to point out that several tissues are affected. Metabolic tissues are affected when you are diagnosed with cancer. So both the liver, the fat, and also the skeletal muscle are affected in various different ways when you are diagnosed with cancer. And we are looking into some of these relationships, both the insulin resistance, also the hyperlipidemia, the inflammation, and also cachexia, which is cancer-induced muscle loss. And the way we have done this in our lab is using the rather common Lewis lung carcinoma model, where you can subcutaneously inject cancer in mice, and then over a period of 3 to 4 weeks, they will develop metabolic dysfunction and cachexia. So a couple of years ago, we published this paper with a bunch of people from the August Krogh section for molecular physiology, using this Lewis lung carcinoma model. And what we saw was that when we, in the black line, we have our wild type controls. In the green line, we have our mice that have a small tumor, and then we have some mice that have big tumors, so a larger tumor burden. And we saw when the mice were exposed to a larger tumor burden, they became insulin resistant. So here we did an ITT, and in our wild type mice, when you give mice insulin, there's a decrease in blood glucose. And this effect of insulin is markedly decreased when the mice have a tumor or have cancer. And in order to see where this insulin resistance took place, we used glucose tracers. So this is gastrocnemius muscle, where we can investigate where the glucose go, basically during the insulin stimulation. We didn't see any effect in basal conditions. And then when you stimulate the mice with insulin, you have increased insulin stimulated glucose uptake into the muscle. But then when you have cancer and you're a mouse, in this case, you become insulin resistant in the muscle. So there's decreased insulin stimulated glucose uptake in the mice that have large tumors here. So, and we are, again, we wanted to know what is going wrong in this condition. And the way we can do this is look at some of the molecular signaling events that happens when muscles are stimulated with insulin. And so we looked a little bit of AKT, which is a part of the more proximal part of it, the insulin signaling cascade. And to our surprise, we actually saw the opposite of what we expected. So instead of decreased insulin signaling, we actually saw increased phosphorylation and activation of AKT indicated here on these two phosphorylation sites. So it's not due to a decreased response at the proximal level of the insulin signaling cascade. But when we talk about glucose uptake into muscle, we also have to remember that there's both, it's affected by both the supply or the supply of glucose in the blood. It's affected by the transport across the sarcolemma on the muscle, and it's affected by the metabolism inside the muscle. So we got help from Kim Sjöberg. He can measure the microvascular perfusion of muscle in mice during insulin stimulation. And this is just the control mice without cancer. And as you see here, that when you stimulate the mice with insulin, there's an increased microvascular perfusion, indicating that this is, and this is also something you see in humans, that there's an increased perfusion of the muscle when you stimulate with insulin. But when we did this in our mice with big tumors, this response was completely abolished. So there's both a defect in the muscle, but also on a systemic level in relation to the microvascular perfusion. So we wondered if we want to investigate what goes on inside the muscle without the effects of the decreased perfusion of the muscle that could affect the insulin stimulated glucose uptake. We went to another model to actually dig into the more intracellular events during the development of cancer and metabolic dysfunction in cancer. And the way you can do this is that when you have your mouse, here again we're using the Lewis lung carcinoma cancer model, you can do an ex vivo muscle incubation system. So you can take out the soleus and the EDL of the mice, and then you can keep them alive in an incubation chamber and basically stimulate with different compounds. You can contract them, you can stretch them, and look at glucose metabolism. And in our case, we wanted to look at insulin stimulated glucose uptake. So that's what we did, investigating both EDL, which is a more glycolytic muscle, and also soleus, which is a more oxidative muscle. And here in the gray bars, you have the wild type mice, or the control mice that does not have cancer. During insulin stimulation, as expected, and this is submaximal insulin, you have increased glucose transport into the muscle, both in EDL and in soleus muscle. So our system works, and this has, of course, been shown multiple times, that there's increased glucose transport during insulin stimulation. And when we then looked at EDL, this is our tumor-bearing mice. In EDL, there was no defects. So it seemed like EDL was not affected by having cancer, and they didn't develop insulin resistance per se. But when we look into soleus muscle, this effect of insulin was almost abolished here. And we saw a tendency to increase basal glucose uptake. So it seems like there is the effect of cancer really depends on the muscle type, at least in this tumor model in Lewis lung carcinoma. And we wanted to dig into this insulin signaling cascade to try to understand. Now we have a system that is outside of the body with without the defects of the microvascular perfusion. So now we can dig into several of these signaling cascades and look where it actually goes wrong. And while again we saw no defect of AKT phosphorylation was not affected, PRES phosphorylation downstream of AKT was not affected. We saw a little bit of changes in signaling on GSK3, but what really was interesting was this protein TBC1D4, which is involved in the GLUT4 translocation to the sarcolemma. And again, our control mice without cancer, we saw a nice increased phosphorylation of this protein, TBC1D4, on several sites. And here, remember that a phosphorylation is actually an inactivation of this. It's a RAP-GAP GGPase. So it's actually, we inhibit a protein that inhibits the translocation. But there's an increased phosphorylation as expected in our control mice. But again, and this is, I'm just showing you data on the soleus muscle where we have the insulin resistance. Again, there's no effect of the insulin on these sites in our tumor-bearing mice. And this is despite having a normal phosphorylation of other AKT substrates. So it seems like there is something what other groups, for example, David James's group in Australia, they have looked into this selective insulin resistance. So it's not the whole insulin signaling cascade that are affected, but it's specific parts. And here, it seems like TBC1D4 is a site that is affected in cancer. So in a short summary before continuing, that patients that are diagnosed with cancer, they do show whole body insulin resistance when you measure with a clamp. And this we can also see in our preclinical models. So it seems like it's a thing that happens on a multiple species level when you investigate cancer and metabolic dysfunction. Then in addition, it seems like it's not, muscles are not affected in the same way here with EDL and soleus, where it was mostly pronounced or most pronounced in soleus muscle. And then we saw some of the defects in a more selective insulin resistance, where it was specific parts of the insulin signaling cascade that was affected by the cancer. But what about AMPK? This was one of the main things of my talk today. So let's dig into this cool protein. So for those that don't know, AMPK is a metabolic stress sensor. So during different stressors like acute exercise, hypoxia, fasting, and also cancer, you can see that there's an increased activity of this protein AMPK. But when you also talk about increased AMPK, you also have to talk about all the mechanism it has been shown to be involved in. So it is a notorious protein to work with because it's involved in so many different processes. And this includes glucose metabolism, protein synthesis, fatty acid metabolism, mitochondrial biogenesis, and autophagy. And this is just to name a few. So AMPK affects many things all in the muscle. But for us, when we look at glucose uptake and glucose metabolism, then if you stimulate AMPK with a pharmacological activator, you can increase muscle glucose uptake in an AMPK-dependent manner. In addition, Rasmus Købsted and Jørgen Wojciechowski, also from Copenhagen, they have shown that the AMPK is necessary for this improved insulin sensitivity you see after contraction. So AMPK is involved in glucose metabolism. And when we then look into cancer, and in this case, cachexia, one study have seen increased phosphorylation, which is an activating phosphorylation site of AMPK. There's an increased phosphorylation in cachectic patients. And we and others, this is the Lewis lung carcinoma, in the muscle, there's an increased phosphorylation and activity of AMPK. And this is not, many groups have shown this increased phosphorylation in the muscle of tumor-bearing mice, and especially cachectic mice. But the function of muscle AMPK in relation to glucose metabolism has not really been investigated. So we wanted to look into this. And first, we wanted to see whether we could actually see the same effect in another cohort or another group of patients diagnosed with cancer. So we, through a collaboration with Raun Langen at Maastricht University, we were lucky to get our hands on some biopsies from non-small cell lung carcinoma patients with and without cachexia. And while the phosphorylation of AMPK was not increased, or it wasn't affected in our group of patients, then we saw that most or all of the subunits, so AMPK down here, it's a heterotrimer. So there's an alpha subunit, there's a beta subunit, and there's a gamma subunit. All these subunits were increased in our patients diagnosed with cancer. So while the phosphorylation was not increased, we did see an upregulation of the total protein of AMPK in these patients. And when we look into the clinical characteristics, if you just focus on at the bottom down here, and for example, the weight loss, so there was a correlation between how much the patient lost during their cancer, or when they were diagnosed with cancer, and the elevation or the expression of AMPK on a total protein level. So it seems like there is an association between AMPK and also more the clinical characteristic of cachexia. So we went back to our Lewis lung carcinoma model, and here we can either remove the activity of AMPK in muscle, or we can increase the activity of AMPK and then investigate what happens when a mouse gets cancer. So we have this muscle-specific dominant negative AMPK, and basically it's a transgenic model where you have overexpressed a kinase that is not active. So you will still have the protein, but it cannot be activated. So it's a dominant negative mutant. And when we do this and we give the mice cancer, or the mice are subjected to cancer, then very early on, so this is at day 14, you actually start seeing glucose intolerance only in our mice that lacks AMPK activity in the muscle, where we do not see it in our wild type wild type mice that are subjected to cancer. And again, looking into our muscles and what goes on in the muscle, if we just focus over here, this is our tumor-bearing mice. So we have increased insulin stimulation in our wild-type mice, but when you lack AMPK during cancer development and increased tumor burden, you actually become, or the mice become, insulin resistant, specifically in the muscle. So AMPK seems to have a more protective mechanism, because when you lack AMPK, the mice are worse off during cancer development. And we talked about this protein TBC1 before, and here is the phosphorylation by AKT. And in our control mice, nothing happens when you lack AMPK activity in the muscle in relation to the phosphorylation during insulin stimulation. But, and this is where it becomes a little bit tricky, in our wild-type mice, just like we saw with the AKT phosphorylation. In our wild-type mice, when they're subjected to cancer, there's an increased phosphorylation on TBC1D4, fitting with the increased AKT phosphorylation we also observed in our previous studies. But in our KD, or in our mice lacking AMPK in the muscle, this increase does not happen, basically. So we think that AMPK might be involved in some of the more protective mechanisms there are in the muscles when the mice are subjected to cancer. And due to the sake of time, I can't go into this, but we also saw that AMPK are likely regulating the expression of PDH and PDKs in the muscle. But I can't get into this due to time, but of course, you can read the paper when it's out, hopefully. Then we could do the opposite, so we can activate AMPK in the muscle, and hopefully maybe we can prevent. That was our hypothesis, at least. We can prevent some of the insulin resistance you see in this model. And when here you have our vehicle, our control mice, and then we have in the gray dotted line, you have our LLC, so our wild type tumor bearing mice that are non-treated or treated with a vehicle. And then we treated our mice with LLC with ICA, which is a known AMPK activator that activates AMPK everywhere. And we actually saw when we look at the incremental area under the curve, that while the mice become insulin resistant as expected, when we treated our mice with ICA for chronically, I should say, this is they are treated after 7 days after the inoculation of cancer, and then the remaining 2 weeks they are treated every day with ICA. And then you see this improvement of insulin tolerance. And in addition, just to show some of the molecular stuff that also goes on in the muscle, while we see this decrease in phosphorylation of mTOR signaling that is involved in muscle hypertrophy, this is rescued when we treat our mice with ICA, again suggesting that ICA may have a protective mechanism in cancer and development of cachexia. So this is the overall story of this AMPK, or the involvement of AMPK in cancer-induced metabolic dysfunction and cachexia. And this paper is on bioRxiv, and we are currently doing revision as well. But until then, you can read the paper on bioRxiv. Yes. So to sum up, we have in the group of University of Copenhagen here, where I'm at doing my postdoc, we've seen that there's development of selective insulin resistance in tumor-bearing mice, and it seems like TBC1D4 is involved in the development of insulin resistance. In addition, we've also seen that AMPK might have a protective role against some of this cancer-induced metabolic dysfunction, because when you lack it, the mice and the muscles are worse off, while if you activate AMPK chronically over several days, you actually improve or prevent some of the insulin resistance in tumor-bearing mice. And with that, I will say thank you to all these people involved, especially Lykke's group up here. I wouldn't be able to do this without all these amazing people, my family, the group I did my PhD, and all of my other friends. So thank you, and I'll be happy to take any suggestions, questions.
All right, thank you very much, Steffen. So usually a perk of being the chair is that I get to ask the first question, but I can see that the chat is already going. So for those of you who wants to ask a question to Steffen, you can type it in the chat, or even better, turn on your cameras and ask it yourself. So if you want to ask the question, just raise your hand or just turn on your camera. But otherwise, I will read out the questions in the chat. Yeah. All right, so I think we'll start with the first question from Baptist Jude. In the tumor model, do you have less glucose perfusion in the muscle, and or the glucose doesn't cross endothelial barrier responsible for less glucose uptake in vivo?
Do you have less glucose perfusion in the muscle? So we did see during in our tumor model, there is less microvascular perfusion in the model or in the muscle. Whether there's a change in the interstitium, so going from the blood to the interstitium to the muscle, we don't know. That is something you can investigate in humans, and I don't know. I think we've tried to set it up in mice, but it's not working 100%. So there's still some optimization to do. And the other part, the glucose doesn't cross the endothelial barrier responsible for less. Yeah, so we're unsure about that step in whether it's across these pores that goes into the interstitium. We don't know. but we can see that at least the perfusion is affected and also the glucose uptake. What goes on in the interstitium, we don't know, unfortunately, but a good question.
Okay, thank you, Steffen. Then it's Samane, and do you want to ask the question yourself, or?
Yes, of course.
Okay.
Yeah, thank you for the great talk. My question is, as a downstream target of AKT and AMPK, when you saw that the phosphorylation didn't take place, did you also see changes in the GLUT4 translocation?
Thank you. That's a good question. And we actually wanted to look at this, and we do have the method to do it, but we haven't measured GLUT4 translocation to the sarcolemma. You could do it with a MUCTAC, but we do have some preliminary data in vitro that suggests that the GLUT4 translocation is also decreased in an in vitro setting. But there's a PhD student in the group I'm in that's looking more into this in cells. But in this mouse model, we haven't investigated GLUT4 translocation per se, but total GLUT4 is not affected.
Oh, okay. Thank you.
Thank you for the question.
Okay, and then I think there was a question from Henning Lange.
Yes, thank you very much, Luca. Great talk, Steffen. I was impressed to see that you got a really good phospho P70S6 kinase signal in your samples, which I know is elusive to get without any should I say anabolic surrounding, without any. So remind me, was the last sample from a lysate, or was that ex vivo? The Western blot for the P70 S6 kinase.
P70, everything I showed here was in vivo. So
that was taken out of the mice.
Sorry?
That was taken out of the mice without any insulin injection or without any.
This was insulin stimulated. So all of our mice were insulin stimulated, and that's also why we can get the P70. Because if we just take basal samples, we almost have no P70S6 kinase signal.
Same. And have you had the chance to also look at any sort of physiological protein synthesis readout, like pure myosin incorporation or stable isotopes or something like that?
This is actually something we want to do, and we want to do it both with amino acid tracers, but also puromycin. We have not yet done it, especially because there is this. There are James Carson, his group, they're looking into AMPK in a different role, that it might be that AMPK is actually inhibiting mTOR and thereby causing cachexia and muscle loss. So it is actually something we want to investigate, because AMPK could inhibit mTOR and then decrease protein synthesis. And that I can't answer that. We haven't done it, but it's a very good question, and we want to answer it at some point when there's time for it. But yeah,
looking forward to it.
Yeah.
All right, looks like I get to have my question now then, Steffen. So we know that there's a little bit of discrepancies between the mouse data and the human data regarding AMPK. And some groups see a marked increase in AMPK phosphorylation in cachectic muscle. We don't see it so much, and we definitely don't see it in the human cachectic muscle. So do you think the discrepancy could be due to sort of the regulation of the different models of the upstream kinases of AMPK, like LKB1 or CAM kinase? And have we looked, or have you looked at that?
We have not looked upstream. One of the things with LKB1 and some of the more upstream of AMPK is that at least LKB1 is constitutively active. So it's always active. And what's really activating AMPK is so you first need the change in AMP and ATP ratio. And then the LKB1, when there's a conformational change, then the phosphorylation happens and there's increased activity. So it could be interesting to see whether AMP and ATP, and it's very hard to do in muscle because it changes so fast. Whether that's actually the cause of the different responses different people see in different contexts and cancers. But it's very hard to do because AMP and ATP switches so fast. But I mean, LKB1 could also be interesting to look at. We haven't done it, and also the CAMP kinase. It's a very good question.
Yeah, I'm wondering if anyone has tried to induce cachexia in any of the knockout
The LKB1 knockouts?
models.
I don't think so. Yeah. But LKB, but I think you run into the same problem with LKB1 affecting so many downstream targets as well. It's not just AMPK. So it is. It's molecular biology is just too complex sometimes.
So another question I have is like, because the AMPK of course can sort of feed into mTOR signaling, but it also feeds into fatty acid metabolism. So do you think that could also play a role in your studies? And if that, do you think that's the major role, or do you think there are other indications that other pathways are important?
Yes, I definitely also think that fatty acid metabolism is involved in cachexia. Again, there's different data suggesting different things, whether there's an increased IMTD, intramuscular triglycerides, in cachexia as well. But I definitely think that if you have issues with the metabolic sensing and your muscle is basically under stress all the time, I think you would have a hard time to actually regulate the switch between glucose metabolism and fat metabolism. So I do think that it's a very valid question to ask, and I think we could also do it, but I think it should also be investigated, especially with this fat muscle crosstalk in cachexia, where it seems like the fat are actually communicating via the bloodstream to muscle and inducing cachexia. So for sure, I think fatty acid metabolism is also involved, and maybe also in our model, but we haven't. I don't have enough data to say either or. I think.
Yeah. Okay. Well, I think let me see. I think there's no more questions. So, unless anyone else wants to have a last question, I think that concludes the session. So thank you so much, Steffen, for sharing your published and unpublished results.
Thank you for having me, and thank you for all the good questions.